So let's see how you did on the lecture, right? And make sure that these were all fair things that we had talked about the other day in lecture, right? So workbenches where proteins are assembled, what are the workbenches called? Ribosomes. Scaffolding and highway for the cell. Cytoskeleton. Houses and organizes cellular DNA. Nucleus. Keeps the surface of the cell clear of debris and mucus by moving things past the cell. Cilia. Very good. Where proteins will be produced. Now, the workbenches are the ribosomes, but those workbenches are embedded in an organelle called the rough endoplasmic reticulum. Is that right? Aids in cellular division. It's the only thing I wanted you to know about these. It's the last things we talked about. Centrioles. Contains digestive enzymes, lysosomes. Provides the majority of the ATP a cell will need. Mitochondria. Contains antioxidants for neutralization of free radicals. Peroxisomes. Increases the surface area of a cell for rapid absorption. Microvilli. Where lipids are made in the cell. Smooth ER. Where products made in the cell go for wrapping and shipping. All right, so there were, I think that was with confidence. The majority, it sounded like you guys kind of got that. Do you think you'll see a lot of these on the next test, on the first test? You really will. Squamous cells could be described as what? Thin, flat, amorphous. Maybe you said that, you know, so a lot of different things. You didn't leave it blank. Stellate as star-like. Phospholipids make up most of the plasma membrane. What organic molecule, what class or category, carries out multiple functions of the plasma membrane? Proteins. Did you put it? Celebrate. Celebrate. That's exactly right. Diffusion is the movement of solutes between two areas of concentration, whereas osmosis is the movement of water. How is active transport of solutes different from passive movement? Active requires ATP. Requires ATP. Describe what would happen in a cell placed in a hypertonic solution. What would happen to that cell? It would shrivel, it would shrink, it would crenate. Great. Define edema. It's really an excess of fluid between the tissue spaces and the tissue spaces between cells. So swelling. If you said swelling, that was fine. I mean, because kind of it is. Swelling means it's an excess, right? So were these fair? Y'all were ready? You know, because it's a job now. It's not just like school. It's a job. You have to come in ready, right? All right, so great. And the lab, I was incredibly careful that I didn't put anything on that lab that wasn't in what I told you would be used. I opened up the visible body, I scrolled down the medical terminology, and just took them from there and gave you a couple of extra hints. For the directional terms, the same thing. And then I opened up the book, I looked at our regions of the body and just did the same thing. Filling in the blank. You were expecting it. You were prepared, right? All right, so you all have a test 1 week from today. It is your first test. It will be chapters 1 through 3. That test, as I have stated in the past, a test is all multiple choice questions. These multiple choice questions will come from lectures, and you all studied probably a lot more about the lecture than I ask quizzes on, but can I ask any multiple choice questions about anything from lectures? Yes. Go back and review your lecture notes. They should just be a review for you at this point, right? And what else are on test? The deeper insights. No lab stuff is on a test. The labs are going to be lab quiz. There'll be lab quizzes for labs. So no lab work will be on these multiple choice questions. Let me say this to you, and I think y'all would trust me. I've been doing this a while, right? what I tend to see is that people don't do as well on tests as they do on lecture quizzes. So please, don't you be sitting there thinking that, oh, this is multiple choice, I got this, because maybe you don't. Let me say it again, I've been doing this a long time. I have over 100 students every semester. Usually test grades are lower than lecture quiz grades. So study hard. You need to know this information. Those questions, I feel like my questions on the test, you either know it or you don't, and otherwise you're guessing, right? So make sure you're studying. That test will be 1 week from today. It is chapters 1 through 3. And I do want to let you all know, and I'm not saying this because I think you need to do this. I don't. But I'm just letting you know, just so you know, some important dates for this class. Some important dates for this class, since it's an 8-week class, is that the last day to withdraw without academic penalty, which means that it would be a W, is February 7th. That gives you all plenty of time to use this week, bring your grades up with this test grade. Try to bring them up. Some of you have some ways to go or to figure out how to study. But just try to get where you need to be, right? Do I have any questions about what's expected in this class? You know, it was in the contract that every day you came in, you'd be ready at 9 o'clock for what we've done the day before. That was in the contract that you signed. So do I have any questions, though, seriously? Yeah? All right. Yes, Jada.
So for the test, for most, like, the quizzes that we do,
yes,
is all the stuff that's on the quiz on the test?
Well, maybe not all of it, but a lot of it will be. But it'll just be a multiple choice question about it. But a lot of it will. So if I were you, if I were a student, and I've been where you are, I have been a student in this class, you know, a long time ago. But here's what I would do in a class like this. I'd study everything. I'd study every one of my notes. I'd study those lecture quizzes. I'd go on and use some of my classmates' study guides that they put up nicely for me on the discussion board, because y'all have already figured out which students have those really nice study guides, haven't you? Have you figured that out? If you haven't, you're not really a savvy student yet. If you haven't been using them, then you've got some things to learn about how to get through a class like this. I'd use everything available to me. I'd read the chapters before the lecture happened. I would go back and listen to lecture. I mean, that's just what you need to be doing. If you don't have the grade you want, that's what you need to be doing, right? But yes, I would definitely use the lecture quizzes. The reason I would use them is that you know that I have thought that what was on the lecture quiz was important enough to quiz you on, that you will likely see that in a multiple choice question on the test. It's a good question, Jada. It's a good question. So yes, use those and use everything else too. Will there be a single question on that test that wasn't what I told you to study? No. So you know how to study. You study your lecture notes and you study the deeper insights. And you study your lecture notes if they're really good notes, right? Let me tell you what I saw the other day. I think it's important enough that I let the whole class know this. I had, you know, you can take classes on how to take notes in class. Sounds kind of silly, doesn't it? But you can. And I think it's a really good idea to Google how to take notes in a class like this. Here's what you don't do. You don't copy what's on a PowerPoint verbatim. You don't do that. It's on the PowerPoint. You could always go read it. You're listening for inflections. You're listening for how something's paraphrased. You're listening for how something's applied. And you're grabbing that. That's what you're doing. Your notes are just usually, should be in, no one else should be able to decipher them but you, right? And then you can actually, because you're not writing longhand, right? And then you're going back and you're using lecture. Not all classes have lecture, but you're using the recording. Or you're using your book, or you're using Google or you're using something to make those notes you wrote make sense, right? So please don't spend your time in here with me that you're not actively engaged with what we're talking about. I've had a couple people, and I haven't even called you out on it, but don't think I haven't seen you, that have your phones out. That's one of the worst things you could do in here, is to let somebody divert you from what's going on in here, right? So, okay. Okay. All right, so here we go. Let me tell you about today's, what's going to happen today. So today, we're going to start chapter 4. This will not be on the test next week, this information. This information will be on a lecture quiz Thursday. So you will expect it on a lecture quiz Thursday. So you will have studied it for that. But I really don't expect you, from the lecture that we have Thursday, I don't expect you to be ready for a lecture quiz on that Tuesday when you have a test that you're coming in for. So is everybody clear that when you have a test scheduled in here at 9 o'clock, you will not, that's the one day you will not have a lecture quiz? Any questions about that? But since we are starting new material, I do expect you all to stay up with it. It's the only way so many people can pass. So stay up with it and be ready for this lecture quiz on Thursday, okay? I do understand that probably after Thursday's class, you all will devote your time to chapters 1 through 3, notes and insights. I get that. So on Tuesday morning, that's all I'm expecting from you is that. Okay? Good? All right. So here's what we've said. We have a foundation now of what is an atom. We understand why and how some atoms play or not. We understand what those bonds are called when they come together and play or not. We understand that some of these molecules end up making macromolecules like DNA and proteins and carbohydrates and lipids, don't we? And they're used to be the building blocks for organelles inside of our cells, right? And we've even seen that not all cells are alike, that some cells have different anatomy because they're going to end up having different functions. Is that right? So what we are going to be getting to here in the next little bit is going to be tissues, because what are tissues made of? Cells. Is that right? But before we get there, it has been really kind of maybe even drilled into you that all disease is at this molecular and cellular level. Isn't that right? And that the cell is alive, that the cell is needing to do work, and the work that it's doing is going to be very prescribed by which molecule? DNA. And how that cell is going to function is really going to be controlled by the cellular DNA. I think we need to take a little bit of a deeper look into that cellular DNA, right? So we're going to do that in chapter 4. We're going to look at how that DNA is functioning. There's some very specific things that I'm going to want you all to know about how DNA dictates what's going on in the cell. We're going to have some lab exercises later today, this morning, early afternoon, that hopefully will help you all to picture what's going on inside the cells. Even we're going to look at some under a microscope and start our look at cells, right? Because can we see the human cell with the naked eye on gross anatomy? No, we can't. We need the aid of magnifying lenses. We need the aid of microscopes to be able to see human cells. So we're going to do all that today. We have a lot to do. I hope you all are excited about it. So let's get started with our understanding of really what's dictating what's going on inside of the cell. We said that the DNA molecule, deoxyribonucleic acid, is the signature molecule of all life, because it is truly dictating everything that's going to happen in the cell and what's going to go on. So we're going to try to get a glimpse of how it is doing that. I told you all, if you saw DNA in this format, what does this look like back here, back in the background? What does this look like? A little what? It looks kind of like a little X, doesn't it? It looks like, and it's attached at a certain little part here. But if we started to unwind this DNA, what did we say what does it look like? It looks like a double helix. It looks like a ladder that's been twisted, doesn't it? And we see that this DNA molecule, just to stay, not to get, so it doesn't get tangled, it looks like it's actually been wrapped around these molecules here. These are called histones. They're proteins. They're histones. And these little histones are truly keeping the DNA from tangling. Can y'all see it in this illustration? Can you see that in this illustration from where you're sitting? Aaron, okay, I mean Nicole, not Aaron. Nicole, that's what I do, okay? So, and that's where a lot of these wrinkles came from, so I would suggest maybe getting those anesthetics. But anyway, so you guys can see how this is. So we're going to take a look at it. I've already given you the distinction between chromatin, when DNA is in its single, it's double-stranded, but it's not in duplicate form, isn't it? It's kind of all stretched out. But this wouldn't be called DNA as chromatin. What would this be? A chromosome, right? And you all said humans have how many chromosomes?
26.
46 total. So 46 total, but we don't think of them that way because they're sexually reproducing organisms, humans are. So we get half of our DNA from where? And half from where? And so we think of them as pairs, don't we? And we have 23 pairs, which is a total of 46, and that's true. Now, here's what I need you all to sort of just to history. It's very little bit of medical history, I have you know, but I do think it's important that you know of a little bit of the history of this. It was only in the early 60s that we understood this molecule, what it looked like. And you all just described what it looked like to me. But some of the scientists responsible for this were Watson and Crick. They're the ones you really hear about. So Watson and Crick. But there were many other scientists that contributed. Watson and Crick really got all the fame and the glory and everything from this, understanding this DNA molecule. But they did it by breaking into Rosalind Franklin's lab and stealing her research. Who says crime doesn't pay, right? But no, actually, they really did do that. But here's a problem. Franklin probably shouldn't have been hoarding research, because in science, what should we be doing? Sharing and dispersing, right? She ended up dying very young of cancer, and part of the reason for that was because she really was exposed to a lot of radiation in her research in her lab, so it's sort of a tragic kind of thing. I think you have an insight about that, and it is fascinating. But guys, when you all think about what's going on in medicine today, from infectious disease all the way to metabolic disorders, it is only because of understanding that DNA molecule and the structure of that and genetics that we understand all that we're getting to do today. So it just absolutely blew wide open medical research. In understanding this, medical research took off. And we are where we are today, really, because of understanding this DNA molecule and what it looks like. So this says chromatin, and y'all know chromatin is like this, but this DNA is now in this duplicate form, and it's condensed. And so we call those, we call that chromosomes, didn't we? All right, so here's what I want you all to know are some basics. You have PowerPoints, you have your textbooks. Listen to me. I'm breaking it down for you. Trust me on this. I am. I'll tell you, I had 2 Virginia Tech majors summer before last, biology graduates from Virginia Tech, who said they never really got this because they had memorized, and I'm sure they were really brilliant, but they had never really gotten this because they never really bothered to break it down. But I am breaking it down for you, so listen. All right, the DNA molecule. DNA dictates all the diversity we see, even with one species in this room, and room 29 at SVCC, Homo sapiens, look around at each other. DNA is dictating how those atoms are going to make the molecules and how things are going to end up producing proteins. And look around at the diversity. Any 2 people in here with the same skin tone? Exactly the same eye color? No. Hairlines? No. Fingerprints? No. Everything, everybody, well, fingerprints a little bit different, but anyway, No, all the diversity, and this is just one species, right? And DNA is dictating every single thing. Now think about all the species that have been on the planet, okay? Think about all the species that are on the planet today, that are living today. So think about all the different, just within the animal kingdom, all the diversity. And what did you say is dictating that diversity? DNA. So DNA must be incredibly complex. The molecule must be incredibly complex. Thank you, Watson and Crick and Franklin, right? Roll. I want you all to think about DNA, and I am going to take a ladder, because y'all said it's like a ladder with rungs, didn't you? And I'm going to lay it on its side. You with me on this so far? And I want you to just think about right now the sides of the ladder, if you will. And I just, because of the difference in DNA dictating all this diversity and all the different traits and characteristics that we're seeing, it's got to be complex. Wait, no. There's a phosphate group here and a sugar. Phosphate, phosphate, sugar, phosphate, sugar, phosphate. Are we bored yet? If you're not bored yet, you weren't listening. Phosphate, sugar. Guess what comes right here? What comes here? And will we just keep going? Okay, well, that strand is not complicated. It's got, the complexity has to be in the other strand, right? It must be in that complementary strand. Oh, wait, guess what's here? And guess what's here? Guess what's here? Guess what's here? Are you kidding me? Are you kidding me? So, by the way, what is the sugar? What is the sugar in deoxyribonucleic acids? Deoxyribose. Deoxyribose. How did I know that's the sugar? OSE tells me it's the sugar, isn't it? So, okay, so far I know that the rungs of the ladder must be where all the complexity comes in. I'm sweating just thinking about how complex this must be. Is that right, Alexis? The complexity in the rungs? Look at the diversity you see. The diversity that you're seeing is from DNA, and you're telling me it's not complex in the rungs? Let me tell you about DNA. DNA, and this is in all things, your bacteria that are in your skin and in your colon and in crevices, your proteins, your yeast, your pets at home. The DNA looks like this. In the rungs, there are 4 bases. These are called nitrogenous bases because guess what they have? Nitrogen. That's why they're called nitrogenous bases. And there are 4 bases. And for now, I just want you to abbreviate them, but you will need to know what they stand for. Oops. You will need to know what they stand for. Adenine. Really? Adenine, thymine, cytosine, and guanine. And yes, you need to know what they stand for and how to spell those 4. Okay, so is everybody just listening to me now? They're just listening and trying to picture this? So I'm telling you that all DNA for all species in the rungs of the ladder, there's just these 4 bases. Is that right? So there might be an adenine here, and there might be a thymine here, and a thymine here, and then there's a cytosine, and then there might be a guanine, and here there might be a cytosine. Are you serious? That's it? So if I were to go back to that coil, double helix coil, that's it? On this other strand, I want you all to know that there's something we call the law of complementary base pairing. Y'all write that down, the law of complementary base pairing. Did you write that down? And let me tell you what the law of complementary base pairing says. It says that if on this strand there is an adenine here, it can only base pair with a thymine on this side. If there's a thymine here, it would only be pairing with the what? So adenine to thymine and cytosine to guanine. So y'all tell me what's on this side? Adenine. And what's right here? And what's right here? All right, some people are answering. Is everybody getting it? What's right here? Are you telling me that our DNA, if stretched out, would be about 6 meters long, that's as complex as it is? That's it? Well, what gives all the diversity? What gives all the diversity? If that's all there is to this molecule, what is giving the diversity? The sequence of the nitrogenous bases. That's exactly right, Andrea and Ashley. It's the sequence. So what gives us our diversity is the difference in the sequencing of these bases. But also what gives diversity is how much you have, right? How long, how many chromosomes you have. Some species don't have 23 pairs. Some species have more. Some species have less. But is the molecule itself any different than that? And what's given all the diversity is just the different sequencing of the 4 bases. Would that mean that the other side is actually different? Or what would we go to know what was on the other side? What law? Base pairing, which will say that wherever there's an adenine, there's going to be what on the other side? Wherever there's a thymine, obviously, that means there's an adenine on the other side. Do you get that? Do I have any questions about that? Is that even tough so far? Really? All right, now I need to tell you all this. Instead of drawing like a line between the sugar, what was the sugar that you said? Deoxyribose. It's a ribose that's missing an oxygen. It's deoxyribose. I saw these lines here, and I've got lines here because the bonding is actually a covalent type bonding. But I didn't draw real lines in between these two because guess what? This is just an attraction holding those rungs, holding these bases together in the rungs. And what kind of attraction is that? What kind of chemical bond did I tell you is the weakest, but the most important physiologically? It's a what? Hydrogen bonds. Holding these 2 strands together. Can you picture it, Luz? Can you picture this DNA molecule? Can you? Julie? Do you have any questions about what I've drawn? No. No? You feel pretty good about that? You know this is like a ladder that's been laid over, right? And the sides of the ladder aren't complex at all. Phosphate group, sugar, phosphate, sugar, phosphate, sugar. But in the rungs of the ladder are these 4 bases arranged like this. Good? All right, now let me tell you all something. And this is true. And we could go back to that picture where y'all are seeing it unwound. Recipes for your traits. Did I tell you DNA molecules hold the recipes for all your traits? Did I say that? The recipes are only going to be on one of the strands. And right now the book is what? Closed. To be able to see a recipe, what do you have to do to your recipe book? You have to open it. Is it going to be hard to open these 2 strands? Because what kind of bonds are keeping them together? Hydrogen, so it's not hard. They will readily open where they need to open for recipe to be read. Are you good with that? It does take enzymes to do it. Enzymes make things happen fast, right? But you don't have to know that. I'm just telling you that they will readily open. Now, will you all bear with me if I have to erase something so you can see the next step? Here's what I want you to realize. The recipe for a protein or trait, characteristic that we're thinking of is going to be on this side. So this side has to get out of the way, has to be opened up and get out of the way. So you all understand that when these hydrogen bonds break, this moves out of the way, but it doesn't really disappear. Does it disappear? No. It just, I'm making it disappear because I need to show you the next step. But it just moves out of the way, and it's hanging down here. Do you see it? Do you see it? Yes. Okay, good. I'm glad you see it because it's down there. It is. It didn't break up. It just moved out of the way, just like a book opening for a recipe. if you open a book for a recipe and the recipe's on this side, it's not like this side disappeared, did it? No, it got out of the way so you could read. So guess what the side that holds the recipe is called? It's called the sense strand. Because guess what? It's going to make sense. This is going to be the one that actually has the recipe for this particular protein. Guess what the one that's hanging down here is called? It's called the nonsense strand. It is. That's what it's called, the nonsense strand. And we know it's down there, don't we? Do we know it's down there? Tell me where the DNA is inside of a cell. It's in the nucleus. So where is this all happening? In the nucleus. I need for you all to know that to get from DNA to a molecule of protein, and I haven't said a single thing about amino acids yet, have I? But to get from DNA to protein, which I told you is the same as saying from getting to DNA to a trait, or getting from DNA to a characteristic, didn't I? To get to DNA to protein, there are 2 steps that occur. Do you think I need you to know those 2 steps? Yeah, and here they are. The first one is called transcription. Transcription. If I were to transcribe something, what did I do? I wrote it, didn't I? I wrote it. That's exactly right. So if I were to transcribe a recipe, I'm going to be writing that recipe, aren't I? Am I? All right, so the first step is called transcription. Where is it happening in the cell? In the nucleus. What is the blueprint for the recipe? The DNA, the what strand? That's exactly right. Anything too tough so far? Nothing. I don't think. Everybody knows me in my whole crazy world, but how about y'all? Are y'all good? Maybe? Are you laughing? Okay. Well, laughing. All right. So what is going to be written in this step? This is called transcription, so something must be written. Let me tell you what the writing is going to be. What's going to be written using the sense strand as a blueprint to write it is a molecule that is single-stranded. That strand has a phosphate and a sugar. And oh my God, are we doing this again? A phosphate and a sugar and a phosphate and a sugar and oh my gosh, are you kidding? What's here? And what's here? And what's here? Okay, so far the writing's been pretty easy for me. All right. This is a single-stranded molecule, and it's called RNA. It's actually a special kind of RNA with a little m. It's called a messenger RNA because guess what it's going to be holding? A message. And the message is the copy of the recipe. It is. Do you get it? So in writing this molecule and and by the way, a phosphate and a sugar and a base comes up at one time, one at a time. These are coming. It's being written. It doesn't just happen willy-nilly. It's being written using what as the blueprint? The sense strand of the DNA, right? Let me tell you about RNA. RNA has 4 bases too. RNA has 4 bases too, but RNA does not have thymine. It does not have thymine. It has adenine, it has cytosine, has adenine, has cytosine, has guanine, but it doesn't have thymine. Instead, it has uracil, uracil. So would you know the 4 bases of RNA? Okay, it has uracil. Let me tell you what RNA is different too. You said DNA has how many strands? 2, because the other one is where? Down there waiting to come back and close up. But it's down there. RNA only has how many? 1. RNA, instead of holding every single recipe, this molecule, every single recipe, RNA molecule is only going to have what? A copy, a message of 1. So RNA is small, RNA is single-stranded, right? RNA doesn't have thymine, it has what? Uracil. So instead of there being a thymine here, what do you think might be here? A U, a uracil. What might be here? What is here? Adenine. What's here? What's here? Do y'all get that? What's here? That was a G, by the way. So this is a C. What's here? How did y'all know? How did y'all know how to write the message? The law of complementary base pairing. And how is this being built? What kind of attraction is this here? Hydrogen attractions. Holding it on until it can all be written. And then the message is written. And then does it just stay there? This is the first step. What was this step called? Transcription. What was happening? What was built? A messenger RNA. What was it? What was used to build it? The sense strand of the what? DNA. All of this took enzymes, catalysts for this to happen, and it certainly took the building blocks, phosphates. The sugar, by the way, the sugar is not deoxyribose. The sugar is what? Ribose. So there's some differences in the RNA molecule from the DNA molecule, but they're both nucleic acids. They both have nucleotides where they have a phosphate, a sugar, and a base. Phosphate, sugar, and a base. That's what a nucleotide is, right? And they're composed of many nucleotides. But which one is the bigger, has the bigger number of nucleotides, DNA or RNA? DNA has billions of nucleotides, where RNA is, this messenger RNA is just one recipe. It's tiny. If this were the end of the story, oh my gosh, nothing would be happening in the cell. This is not the end of the story. This is the first step, which was what? So it's dotting to know the second step. By the way, why do we care about these steps? Why do we care about knowing this? What you say?
It's the beginning, where it starts.
It's where all of the instructions for the cell, to understand the cell, you have to understand it. This has to happen for anything that goes on in the cell. Because cells are essentially protein factories. What's being this the recipe for? A protein, a trait, a characteristic, right? Is that right? And by the way, some diseases interrupt these one or the other of these two steps. Would you like to know which one is which? Yeah, and you'd have to understand that, wouldn't you? Some medicines that are given actually inhibit one of these steps. Y'all have learned one step. What was the step? Who wants to know really the end of the story? It's no more complicated than this. Who wants to know the second step? Do you?
Yes.
Okay. I would hope you'd want to know at this point. Okay. So here we go. Don't write this down. Just see if this doesn't make sense. We're in the nucleus right here. But you all have already told me that's not where proteins get built. You told me they get built in what? Workbench, which is embedded in what? Rough ER, but some of them are free too. But it has to go to a workbench, doesn't it? You all told me that all of that is not happening in the nucleus. It's happening out in the cytoplasm of the cell on those organelles. And then you told me once those things get made, they have to get wrapped and shipped, right? And you also sort of get the idea that this takes some work. Is that okay? Because do you have charged batteries? Yeah. All right. So let's see what happens next. Would it make sense to you, since you told me this isn't a tightly held bond, would it make sense to you that this will unlaunch from the DNA? So I want you all to picture this whole molecule, this recipe, unlaunching from this DNA. And you know the DNA doesn't go anywhere, even though I'm erasing it, right? We know it's still there, isn't it? Along with its complementary strand down here. But this is going to unlaunch. And when it unlaunches from the DNA molecule, this small single-stranded molecule will move out of the nucleus through those nuclear pores and go to the where? To the where? It will go to the workbench. Do you see the workbench it just went to? Right? Let me tell you about this workbench molecule. One of the reasons it's kind of attracted to this is that this workbench, which is huge, this workbench is a special molecule of, guess what? It's a special molecule of RNA, but it's called ribosomal RNA because it is making up the workbench, which is the what? Ribosome, right? So here we've got, I see that. I saw that happened. Did you see it? Did you see the messenger RNA unlaunched for the DNA and then moved out of the nucleus through those little pores and came to a workbench? And so now here it is. You ready for the second step? The second step is called translation. When you all hear that something is translated, what does that mean to you?
In terms you understand.
It's been interpreted, right? It's been interpreted. It's been translated from one thing to another. And language, that means you've taken one language and you've translated it to another one, right? A completely different one. This second step of translation, essentially what is happening is we're going to take a nucleic acid. Which nucleic acid is it? Messenger RNA. And we're going to turn it into a protein. That's going to be a good trick. We're going to translate that into a protein. That's going to be a really good trick because proteins have nothing to do really with nucleic acids as far as their chemical structure. What do y'all know is the monomer of a protein? What's the monomer building blocks for proteins? You're supposed to know that. What are they? Can you forget anything you learned before? Or are you bringing this everything? Every day you bring everything with you, right? What did you say it is, Alexis? Amino acids. So how in the world is these nucleotide molecules that are all stranded out here into a nucleic acid, how is that going to be translated into amino acids? How many different amino acids did we say we have? 20 different ones. Is that right? And what did you say is that we are really concerned about in a protein, the beginning of a protein, is those amino acids where they come up and get in line. Is that right? What did we call that? We call that their primary structure, didn't we? Their primary sequencing, their primary structure. So in translation, we're about to figure that out. Are you excited? You should be excited. Are you hot? Because it's also hot in here. Okay. Are you excited about this stuff? Okay. Look. So here's what's happening. Listen to it first. Listen to it. See if it doesn't make sense. And then ask questions, okay? Because I'll go through it again, so you'll be able to write this down again. To really understand translation, how this messenger RNA comes to a ribosomal workbench and gets somehow translated into a primary structure of amino acids, it's going to take a third type of RNA that we need to know about. That third, don't write this down, you listen. That third type of RNA that we need to know about, that third type is called transfer, a little t, RNA. Transfer RNA is actually, that little t stands for transfer, but you could think of it also as tiny, because little transfer RNAs, how many bases do they have? They're tiny. They only have what? 3. And you understand that the DNA molecule that had the original blueprint was billions of nucleotides long. And maybe you understand this messenger RNA is pretty darn long too. It's a lot of these. And they keep going. But the little transfer RNA only has how many? 3. And what do you think the strand is? Oh, wait, a phosphate, a what?
Sugar.
And that phosphate's there. And then a sugar, and then a phosphate. Okay, so the strand isn't exciting. But what do you know might be here? An A? You think that's adenine right here? And what do you think this one is?
T.
Do you think, no, what? There's no T in RNA, so it's a what? U. And what is this one? U. And you all say that that's the only type of little transfer RNA that would fit right here. Why couldn't a little transfer that looks like a CCG come up there? Why couldn't a CCG have come up here? Because we have CCGs. What did you say? Because of the law of complementary base pairing tells me that this is the only sequence of 3 that could come there. Is that what you're saying? And which one would come right here? What is the only one that could come right here? See what? That's the only little transfer RNA that would be able to come there because of the what? The law of complementary base pairing. Now here's what I need to tell you. These little tiny RNAs, the ones that are AUU, they hold a particular amino acid, a specific one, and not any others, just that specific one. And they're holding on to it is why they're called a what? They're holding on to this amino acid. And guess what this one is holding on to? An amino acid. And guess what happens when this amino acid gets brought up to the workbench and gets really close to this amino acid? Guess what will form between them? A peptide bond. And guess what will form between the next one that comes up? And the next one. And the next one. And it's only particular in which way, which transfer RNA will come up because of the what? Because of the what? Tell me. The law of complementary base pairing. Is that true? Could that be true? Is that true? All right, so here's what I want you all to see. I want you all to see this. I want you all to see that if we looked at ribosomal RNA and we knew the first letter, A, U, U, this tells us which amino acid. It's the only amino acid. It can't do any of the other 19. This is the only amino acid that can hold. What was the other one? CGC? CGC, is that what we said? CGC? Did we say it?
And the second row all the way to
Is it-
Go all the way to the 4th.
CG, sorry, I should have been able to do that. Arginine, so because all I had to do was look up to the second letter and the third letter. And are you saying that this is the only amino acid that transfer could be holding?
Yes.
Really? Here's what I want you to know. There are 4 bases. And when we think about these 4 bases, these 4 nitrogenous bases, and I had to do this in a biology class. It was the biggest waste of my life. I can't get that time back. So I'm not going to make you all do this, but I hope it is intuitive to you. If you only had 4 bases, and I were to tell me how many different sequences of 3 there could be, you would say A, U, C, and it would take you some time, right? If I made you write them out, right? But will you trust me that here they are? And for 4 bases, the different sequences of 3, there are 64 potentials. It's 4 to the power of 3. It's a basic math thing too, but I could make you all write it out, and maybe it really would impress upon you that these are the only ones. But even though there's 64 potentials of 3 bases that could be our little transfer guides, do you understand that there are only 20 amino acids? So what would that also intuitively tell you? That some of them might hold the same one, right? Because there's only 20 of them. But guess what? Here's what I want you to look at. Here's what I want you to look at. This sequence this is the only thing that brings methionine up. It's the only sequence that brings that up. And some of them, there's only 2 different sequences that would bring it up. Some of them have a couple, but some of them only have 1 or 2. Do you get that? Do you get that, guys? So here's what I want to go back to see. So you're saying we could look at that chart, and we could tell exactly which amino of the 20 amino acids, which one was right here. And we could look at the chart and we could see which one is right here. And then 80 amino acids down the chain, we could see which one is there. Is that what you're saying? And then at the 120th spot, we could know which one is there. Why did y'all tell me it's important to know which one is at what site? It's the primary structure which dictates how that protein is going to work. Is that right? And y'all told me that even one being out of order could be lethal. Did you say that? Or could be devastating, right? So we care which one is in order. What dictated which transfers came up? What dictated which transfer RNAs came up to the ribosome? The messenger RNA dictated it. But wait a minute. What dictated the messenger RNA? The DNA. And which step? The first step, transcription. Transcription wrote this, and then translation is understanding that these little transfer RNAs come up and get in line in a very specific way because of the law of complementary base pairing. And what are they bringing with them to the workbench? What are transfer RNAs bringing with them to the workbench? An amino acid. And when one gets close to another one, what will form between them? A peptide bond, and then another peptide, and now you're getting your primary structure of your protein. Are you good with that? Are you good with that? Now, so do you all remember how this messenger RNA was written in transcription? Using what as the blueprint? The sense strand of the DNA. What would happen if on that sense strand of the DNA there had been an inversion or deletion, or there had been a change in the base sequence? Would it have affected how this was written? Would it have affected how this was translated? Yes. Potentially, yes. And did we say it could be devastating? Or it might not be devastating. It might be something that was just benign. Not all changes are going to be devastating, but it's the devastating ones we hear about, right? Some of them are really benign and just lead to variances of traits in the population, like changes in eye color, hair color, that gives the variation in traits that we see. Does anybody know what it's called when those bases on the DNA, remember the original DNA you drew? Does anybody recall that you drew it a certain sequence? Does anybody know what it's called when those bases do change? Does anybody know what that's called? It's called a mutation. So you all might have used the term, I hope you didn't call somebody a mutant, but you might have used the term mutation, but did you really know that it means that one of the 4 bases on the DNA sense strand changed? That's what it means. That's what a mutation is. Some are benign, which means that they're just going to cause variations in traits. Some are adaptive, and which means it's going to help to survive. But many we think of as being what? Deadly. We do, or dangerous, or, going to cause problems. All right, so let me ask you a few questions, then we're going to take a little break. Let me, because this is the majority, this is the hardest part of this chapter. Are you hanging with me? Yes. All right, look. Heading from DNA, it's inside of our nucleus, to the understanding of which and how many and what kinds of proteins are made is 2 steps. The first step is it's happening in the nucleus on the sense strand of the DNA. It's called transcription. What is being transcribed? Say it loud. A recipe, which is a what strand? A messenger RNA. For the second step to happen, the messenger RNA unlaunches, moves out of the nucleus to a where? To a ribosome, which is really ribosomal RNA. A second type of RNA. Is that right? When it gets to that workbench, what is going to be called up? Tiny little molecules, nucleic acids called what? Transfer RNA. Why are they called transfer RNA? What are they holding on to? Amino acids. And when they come up and get in the way that they do, because of law of complementary base pairing, what forms between the amino acids? And now you've got your primary structure of your protein. So what's the end product of translation? Is a protein. Do you get that? The end product of translation is a protein, right? Guys, how many, I want you to know that a set of 3 bases on this messenger RNA codes for a particular amino acid, So a set of 3. Those sets of 3 are called codons. A set of 3 bases. Didn't y'all say it's just 3 bases right here that's dictating which amino acid comes up? Did you say? You guys, a set of 3, I'm going to say it again, a set of 3 bases. See the set of 3? Code up, or called codons. Codons. That code up for a particular amino acid. How many different codons do we have? 64. Did everybody understand why it's 64? Did everybody understand why there are 64 potentials? If you only have 4 bases, the potentials that they can arrange themselves in a sequence of 3, there are 64 of them. And did everybody take a second and look at this chart? You didn't have to really look at it right closely, but you kind of looked at this chart, didn't you? And in this chart, do we have all 20 amino acids represented? And in this chart, do we know every sequence of 3, every codon that would call up that particular amino acid? Do we? From this chart. Do you know what this chart is called? It's called the genetic code. This chart is called the genetic code. So in essence, the genetic code is this. Do you want to hear me paraphrase it? Yes. So in essence, the genetic code is that we understand which amino acid will be called up for every particular sequence of 3 bases. We know that. We know which amino acid is going to be there. And I want to give you something to think about before your 10-minute break. I want to give you something to think about. Science grand, really? I mean, really? Seriously? Don't go anywhere. Wait a minute. Okay, all right. You can turn the page. Okay. Hey, guys. These are 46 total chromosomes. They've been exposed, these chromosomes for this human have been exposed to some fluorescein dyes. And they've been looked at under the microscope and taken pictures of and then cut out. So each of these represent a chromosome in this human, right? Is that right? And have y'all ever wondered like how they figure out where on the DNA the trait is located? Have you ever wondered like how they figure out that so-and-so trait is located on the 20th pair of chromosomes at this particular little site, right down here at this little particular site? Or another trait may be found here, and something that could contribute to it might be found here, and maybe even here. Have you ever wondered how? Because how could they possibly do it when we know chromosomes are so incredibly boring? Chromosomes, all they are is these long strands with 4 bases just changing order, right? Just different things. So how would you figure out where the trait is on the DNA? Scientists, there's some scientists who have spent their entire, talk about focus here. Let's talk about focus. Have spent their entire careers studying one protein. And that protein that they study, it's the protein that they figured out the primary sequence of bases. Now from that, don't write this down, I just want you to get your head around this. But because they know the primary sequence of bases, do they know the potential codons? And if they know the potential codons, can they work backwards to find where that sequence of the sense strand would have been? That's how they figured it out. If that didn't give you chill bumps, you weren't listening. So go take a 10-minute break, okay? Let me pause. Okay, so we're back. All right. So we looked at some similarities of DNA and RNA, but we also looked at some differences of the DNA strand versus RNA strand. So we looked at 3 types of RNA and what it takes for the 2 steps to happen. We said that it takes transcription and transcription happening in the nucleus and an end product being a messenger RNA, translation happening at the ribosomes, and the end product being a protein, right? So we understand that, and so okay. And did we know the law of complementary base pairing? We said the law of complementary base pairing is just simply saying that only certain bases can base pair with each other. In DNA, it's the adenine to thymine and cytosine to guanine. And RNA doesn't have thymine, does it? It has uracil. But we understand that, and it helps us to understand how transcription happens, the law of complementary base pairing, and it helps us understand how translation happens between the codons and the anticodons. And so, but that's fine. Okay, so I want you to have a basic definition for genome. A genome is going to be really, when you think about all the genes of one person, so their entire cookbook. Is that right? So their entire cookbook. So when we think about human genomes, that Human Genome Project, when it was begun, was thought that it was going to take, just years and years. It ended up going so quickly because we thought we were complicated. But what we found out is we really aren't that complicated. We have, roughly, I think it's actually gone down a little bit since we figured it really these notes. But anyway, roughly 35,000 genes. By the way, house mice, not that my house has mice, but a house mouse has more genes than we do. So, yeah. So, humans think it's all about them, but guess what? Kind of isn't. So, now, does environment play a huge role in how genes can be expressed? I've just talked to you guys about this idea. That how we understand cells function and what proteins are going to be made, all the different diversity. Remember, proteins have the most diversity, don't they? And how they're going to get built, and all those proteins carrying out different functions in the cell depends on the DNA, and if the DNA has the recipes for them. And that's really true. But what I also need you all to understand is there's another layer to that, and that study of that other layer to that is called epigenetics. How many of you have ever heard of epigenetics? How many's heard of epigenetics? Raise your hands. No? Not many of you. Okay, so it's a field of study. It's an entire field of study all on its own, and we understand that for this DNA to carry out the instructions for the cell, there are these switches, almost like on and off switches on top of the DNA. You can kind of think of it that way as far as epigenetics goes. And it's the environment that turns on and off those switches. So what you're environmentally exposed to may be turning on switches you don't want turned on. You get what I mean? You guys have all, I'll give you an example of epigenetics. You guys have all heard that some people have the genes for cancer, haven't you? But does that mean they'll develop cancer for sure? No. What has to be turned on? A switch. And that's understanding the DNA has this other level associated with it. It's called epigenetics, where those switches get turned on and off. And are we finding out tons about that and understanding how those switches get turned on and off? Yes. What are some environmental things we know can adversely affect your switches? Smoking is a huge one. Smoking is a huge one. What'd you say? Well, yeah. Well, that's actually, in fact, it's an inheritable trait. Alcoholism, gambling, addictions. How many of you would say, and I'm the first to raise my hand, you had addictions that run in the family? Yeah, right. Whenever I hear that, I think, what family doesn't? But then again, I'm from the South, so I'm thinking like everybody. But there's some families that don't. But anyway, you all know what I mean. And sometimes, it's really difficult diseases to handle. But other things like chemicals in our foods, right? Preservatives in our foods that we're exposed to, those types of things. So anyway, we know they can act as switches. We know that people who are under tremendous stress for long periods of time, whether it be from famine or whether it be emotional stress or whatever, they tend to have more of certain diseases. And again, why? Because of what we know is happening. The switches being turned on and off. So that's epigenetics, and I don't think they talk about it in here, but I feel like students need to know about that. So, all right. So the genetic code. How many different codons are there? How many total possible sets of 3? Or 64. But how many different amino acids are there? Only 20. So it makes sense to us that some of them are going to have different sequences that tell them up. Right? But anyway, great. Transcription, did we know it? We know what it is. Translation, we know. Guys, this is my favorite electron micrograph in your entire textbook, and maybe just ever inside of a cell. This is my favorite one because it really does show us using electron microscopy. We can see inside of cells, and we can see the structure of organelles. And when we see the structure of organelles, does their structure help us to understand how they're functioning?
Yes.
Because structure dictates function, doesn't it? So here, I want to, in case you don't know what this is you're looking at, I'd love to explain to you all what this is, because it explains how cells can do things at the pace they do. This is just FYI. Anyway, so this is inside of a cell. And what you are seeing here, all this grainy stuff inside of a cell, this is the cytoplasm, and certainly there are all kinds of molecules floating around. We know that inside of our cell, it's filled with a lot of things. These are ribosomes. Do y'all see these things? These big things? Those are ribosomes. Now, I want you to, if you can imagine, you can't see it the whole way through, but if you look, there looks like there's a strand. You can't see it the whole way through, but you can look like there's a strand almost connecting those ribosomes. Do you all see that? And what is happening is when this strand got to the ribosomal workbench, this strand is a messenger RNA. And as it got to this ribosomal workbench, transfer RNAs brought up little tiny amino acids that started to do what? Form what? Chains. And as that messenger RNA moved from one, almost like a ticker tape, moved from one ribosome to the next one, chains of amino acids were coming off of them because transfer RNAs were bringing them up. So these are the beginning of the protein chains, right? The amino acid chains. This was the message that's moving through a ton of these ribosomes. All of these proteins would end up being the same protein because it was from the same what? Message. And this is how cells can make proteins at the phenomenal pace that they can make them. Is that the coolest picture? Like if that's, I do get a little crazy about this. I realize I maybe go a little crazy about this picture because at one of my classes, and please don't do this, I'm really trying to get rid of sugar. Let's talk about addictions, sugar addictions. Okay. But anyway, I had a student bring at the end of the semester when it was all done, the final exam. She went out and then she came back in and she had a cake that had this on the cake that had this picture. I was like, she's like, do you know what this is? I was like, I absolutely know what that is. That's like the ribosome picture. So anyway, it was awesome. It looked just like the picture. Anyway, so you guys, we get this idea that we have the DNA, that's our blueprint, right? And from that sense strand, we have the messenger RNA that is going to be written. On the messenger RNA, we're going to have tiny little transfer RNAs bringing up what? Amino acids. And what are forming between them? Exactly. All right. Hey guys, so now when we see a little piece of a nucleus, this is the big nucleus out here, and this is a pore in the nucleus, the messenger RNA comes through, gets on these ribosomes, and as the message is being written, what's being made? Proteins. They'll be sent where for wrapping and shipping? To factory, isn't it? Did I tell you all it's a factory? It's a factory. So, so now, here's what I also need you to think about. The DNA does hold all the recipes for all of our proteins that have to be used. But I also need you to know that for cells to divide, what first must make a copy of itself? DNA. So see if this doesn't make perfect sense to you. You said that DNA is a double-stranded molecule, is that right? So here it is, double-stranded. What would happen if it opened up, right? So it's opening up, and so now there's one strand here and one strand here. We know that this is holding a base, and that's holding a base. But what happens when DNA copies itself is when it opens up this way, each of the old strands will be the blueprints for the new strands to be built on, right? And because that's the case, this strand will end up being identical. The two new strands are identical to the old one. Why? Because of what law? So what you could think is, because some of you are looking really confused at that, but I told you that if you had a DNA molecule and there's an A here and there's a what here? T, and there was a C here and there's what here? D and a C, G and a C, and you get, it would just keep going. If these opened up, what new one would come here? T, and what new nucleotide would come here? G. Okay, but on this old strand, what new one would come here? You get it? And what new one would come here? And is this identical to this? Do you get that? That's called the law of semi-conservation. I don't ever ask you that, but that is what is happening there. I would just want you to intuitively get that if a DNA strands are, the nitrogenous bases are a certain way, and they opened up and new strands were formed on the old strands, because of the law of complementary base pairing, those two new strands are identical. They are. It's beautiful, isn't it? Caitlin, come on. Isn't it? Yes. Okay, thank you. Okay. It is. It is. Look at that. It's just beautiful. New strands formed. And does it take long for that to happen? No. That can happen pretty quick. So here's what I want you all to know about the cell cycles of life cycles of cells. Do cells have a dictated life cycle? Yes. What's dictating it? DNA, right? But they have dictated. Can they go longer than they're supposed to? If they're not supposed to, that wouldn't be a good thing. They have to follow the rules, right? They should follow the rules. So absolutely. Here's what I want you to know about the life cycle of cells. Because we said that some cells will live for hours, but other cells have to live for decades, don't they? All right? So here's what I want you to know, though. There's two major phases of a life cycle of a cell. The two phases are interphase and mitosis. Do y'all see that on here? Well, I don't know. You can't see from where you're sitting, but maybe y'all see inside this. And this, are all this in your book? This is all in your book, isn't it? So the two cycles of a cell are interphase and the mitotic phase or mitosis. Mitotic phase or mitosis. So interphase, if y'all see this little blue thing, interphase is when the cellular DNA is kind of all spread out. It's kind of all spread out. There's growth and normal metabolic activity happening, and you know, all that's the cell's doing what it's supposed to be doing. Carrying out functions it's supposed to be doing. Now look, then the cell is going to get a signal that it's time for the cell to divide. And I want you all to know, if you hear the term mitosis, mitosis means cell division. That's what it means, cell division. What did you all tell me has to happen before a cell can divide? The DNA had to copy itself. Is that right? So the DNA is going to copy itself. It does that actually in interphase. But then the DNA starts to condense. And now it gets into what type of structure? Chromosome structure. That's right, a chromosome. See if it doesn't make sense to you that the functions of mitosis is for the embryo to develop, the fetus to develop, for you to grow, right? This is all needed for mitosis. How about if you stump your toe and knock the skin off your toe? You know, your toe doesn't fall off, does it? Does it heal and repair? Does it heal and repair? It does. Some tissues can repair better than other tissues, but yes, we tend to think about them repairing. And here, then we said also, for this to happen, that we're going to see these different phases. And y'all said that the DNA itself has now condensed. It's doubled itself, and it's condensed. So guess what? When we look at it under a microscope, we can actually see it now. We can see the DNA because it's in chromosome form, isn't it? If you can see the DNA in chromosome form, you're in the mitotic phase or mitosis. The cell is dividing. The cell is dividing. This beginning, y'all are going to see some of this today, by the way. This cell, it looks like you can see these little squiggly looking little worms. Like I will draw it for you like this. Like if this is a cell, before the nucleus and interphase, it's just kind of granular because the DNA is all spread out. It's not in it's not in condensed strands, right? So it's all spread out. But when you start to see all of a sudden these little squiggly looking little lines, really dark little lines like this is the beginning of mitosis. Then all of a sudden you'll go from seeing little squiggly lines that look like they were just all over the place to looking like they're lining up along the equator of a cell. Can y'all see the plasma membrane of the cell where it's defined? And what does it look like the little chromosomes have done? Lined up along the center. This is called metaphase. This is only for lab. I'm not going to ask you all to define prophase, metaphase, anaphase, and telophase. I'm really not, because that's in lab and you do it in a general biology class. And usually, unless you're going into a genetics lab setting, you're never going to need to know it. But you do need to just understand that the cell goes through these phases to divide, and it has to go through these phases to divide. And we're going to see that. We're going to see actual animal cells in mitosis, but they spend most of their life in which phase? Interphase. They spend most of their life cycle in interphase. But when they need to divide, and you can now see the chromosomes, what phase is that? Mitosis, right? So this is metaphase, and they've lined up along the equator. And guess what little organelles are now going to play their role? I think about centrioles as this. Who wouldn't mind standing up? Two people standing up and helping me play this little game. Come on with it. Chris, I can't get you because I need two people that are going to be clones of each other. Come on, Ashley and Andrea. Come on. And I couldn't get you as a clone, Chris, because you're back. So so you guys come here. All right. So first of all, Ashley, you get to be the copy. First of all, y'all don't see Ashley. Come Ashley behind. First of all, this is a DNA molecule in chromatin form. But she's going to make a copy of herself, and as hook up right here, a copy of herself. And so now this is an identical copy, and they're attached. They're attached at a certain location called the centromere. They're attached here, right? And what they have done now, they're really condensed, and there's a copy, identical, right? Now what happens is during mitosis, they're going to line up along the equator of the cell. We're the cell, right? And a centriole, actually turn back to the other one. A centriole is going to be here. Jane, or Kristen, get up there, you're quicker. Kristen, get over there, you're a centriole. You're a centriole. Yeah. See, we're still a cell, we're one cell. Don't stay way over there. Stay way over there, Kristen. Kristen, you know what I think about with centrioles? I'm a centriole too, you're a centriole. Centrioles are sort of like fishermen, and they really do send out a line. It's a protein. But it's like, go ahead and use your rod and send out a line to your little chromosome there. And I hooked you. And now start reeling them in. Now, do you see? So if they were at metaphase, and now stay right there a second, y'all pretend I reeled her in. But then the centrioles start reeling their copies in, and now a copy of each of the DNAs that were identical copies to each other are now toward the polar ends of the cell, and now the cell can start to do what? It can start to pinch in and divide. Did you see how the centrioles reeled their copy in to the sides? So, okay, thanks guys, y'all can sit down now. But that's really true. So what happens is the chromosomes in metaphase line up. Centrioles send out a line to each of the copies, right? The centriole on this side sending out a little line here, this one to here. Then what do they start to do, the centrioles? Y'all are going to see this in lab today. You're going to see animal cells that this is happening. And so this is called anaphase. They're being pulled apart, aren't they? And telophase in animals is called cleavaging. Why do you think it's called that? Because it looks like what? It looks like cleavage, doesn't it? Because now they start to, you can actually see, this is called telophase. You can actually see where the two new cells are going to be from where the one cell had been, right? But now there's two new cells. Nucleuses will reform, and the DNA is now not in its duplicate form, single form, and it can spread out. And now it's in what form? It's in chromatin form. The DNA is in chromatin. You've got two new cells. I do want you to know that cytokinesis means a doubling of the cytoplasm. Because let's think about this, a doubling of the cytoplasm. You're here. How many cells do you have here? Just one. But y'all know that cells don't just have DNA. They have all kinds of organelles, don't they? They have all kinds of organelles. They have all kinds of things they need for the cell. They need fluids. So what's going to happen is in cytokinesis is that whole cytoplasm and organelles, they have to double so that each new cell has what it needs as far as cytoplasmic inclusions. Are y'all good with that? Cytokinesis. Am I going to make you say pro, meta, and are we going to see it in lab? Yeah, we're going to see it. But am I going to ask you questions in here about it? I just need you to know what mitosis means. What does it mean? Cell division. I need you to know where a cell spends most of its life cycle in. Interphase. I need you to know what the definition of cytokinesis is. What is it? Doubling of the cytoplasm so that each new of the two new cells have what the original cell had. I need you to know that mitosis is a type of cloning. What does cloning mean? What's a clone? A clone is an identical copy. It's a duplicate, an identical copy, isn't it? And so mitosis is where you start with one cell, and how many cells do you end with? You end with two. And when you end with two, are the two identical to the first?
Yes.
I want to give you all, and you don't have to take my word for it. But is everybody listening to me right now? Mm-hmm. If you are trying to read your textbook and find what I'm saying at the same time I'm saying it, you are wasting time. If you want to keep doing it, I don't care, because it's up to you to do what you want to do. But I can tell you that it's taking your attention away. So, okay, because honestly, the textbook is always there for you, but I'm not always here, right? So listen and write down notes and whatever, and then go back and try to find things in the textbook, right? Because that's just a way that some people are, whatever. Okay, but it's up to you. So, all right. So timing, we know that it depends. It's different depending on on whatever the cell is going to be doing. Some cells have to divide frequently. Some aren't going to divide. They're going to rarely go through mitosis. Like our central nervous system tissue that's made of cells, those cells rarely divide. That means you should watch out what you do to your central nervous system. Where are central nervous system cells? Brain and spinal cord, because can they replace themselves? No. When they're gone, they're gone. So,
that means it takes so much work, or I mean, is it just?
It's why it's evolved that way, that those tissues have evolved that way, I would have no idea. I know that they've evolved that way, but they're also very protected. So they're in bony structures as well as like membrane bound. So the spinal cord and the brain are incredibly protected for that reason, but they don't do a lot of dividing. I think if you wanted to, not the way scientific theory, but just think about that, it would maybe be because of this. This is important, so I'm glad you brought that up, that question. When do you think a cell is most at risk for mutations? Cell division. And some mutations can be deadly. So these are cells that rarely divide. Whenever you all have heard of brain cancers and whatever cells, you've heard of brain cancers, haven't you? There are different types of brain cancer. They're not of the central nervous system, actual brain cells. They're of those supportive cells that support the nervous system that do divide. But the brain cells themselves don't. And this is, again, why they say, take care of your nervous system, your central nervous system, because they really don't. So in a way, that's good that they're not, right? In a way, it's bad because of some other things that we'll talk about when we get there. But okay, I want you all to know what a karyotype is. A karyotype. A karyotype is a test that gets performed. A karyotyping is a test that gets performed to actually physically look at an individual's chromosomes. Now, does that take a microscope?
Yes.
Yes. But that's what a karyotyping is. So what would be the point of just looking at someone's chromosomes? What would be the point of that?
To see if the child may have something.
Exactly. So that's exactly right, Alexis. But let me tell you how this karyotyping gets done. It can be done on a lot of different types of cells, but cells that are living. So you can take somebody's cells and you can put them in a petri dish and put food in. And as you're feeding them, they will usually divide a few times. Now they won't go on living for a long time outside of that, unless they're cancer cells, which is what happened with the Henrietta Lacks. Or y'all have all heard of Henrietta Lacks and the HeLa cells. But that happens in any cancer cells, not just hers. But a lot of cancer cells will do that. They'll keep growing. But in other people, to get a karyotype, so you can do amniocentesis where after like the 16th week usually of a fetus growing, they're going to go in with a long needle. It's very sharp, doesn't even hurt. They go in through the abdomen, through the uterus, into the amniotic sac where there's amniotic fluid. Pull some fluid off because what's bathing in the fluid? What's swimming in the fluid? The fetus. And the fetus is sloughing off cells that are living. And they'll get some that are living, then they'll feed them, they'll get them growing, they'll get them in metaphase. Remember metaphase? And they'll expose them to fluorescein dyes and they'll take pictures of them. And then they'll use scissors and they'll cut out the chromosomes and match them up with their pairs. That's what they do. That's how they get to a karyotype. You can also do it through CVS, which is chorionic villi sampling, where they'll go through, they can do this as early as 7-8 weeks. They can go through the vagina just using a speculum, a regular pelvic exam, go through the cervix with a little needle and pull out some chorionic cells that have fetal cell, embryonic cells, and they can do a karyotype thing. So there are a lot of different ways that they can get to this. They just need some living cells from a person and they can get a karyotype. Now, the reason that they do it is what Alexis says, is because a genetic counselor can look at this and within a few seconds, just looking at it, tell if the chromosomes look normal. And there are dozens, just from looking at the chromosomes, there are several dozen chromosomal anomalies that a genetic counselor would be able to tell from looking at them. Some of the chromosomal anomalies are really obvious, and it wouldn't take a genetic counselor to look at. Some of them would look at the karyotyping and they would see right away that there's not 46 chromosomes, should there be. There's some chromosomal anomalies where there's only 45. And then there's some that have 46. There's some where a piece of one, because we know in the 20, if you notice, and this was arbitrarily done in the beginning, but now it's the way it's done. If you'll notice that the first pair is the longest pair, so the first through the 22nd pair have been designated by their length. And also where they're held together, where their centromeres are held together. So the first pair is the longest with the centromere. The second pair is the next longest, but the centromere is slightly off center. Do you get that? Mm-hmm. And they get shorter and shorter. And this is the way chromosomes are defined. The first through the 22nd pair, I want you to know, are called autosomes. I think I told you that. That's self chromosomes. And the 23rd pair has been designated as sex chromosomes because one of the many traits that come from that are going to be gender. Okay? So that's why they're called that. Now, this is called a karyotyping. And so we're gonna do some karyotyping in lab. Y'all are gonna practice karyotyping, but this is really, really valuable. Some of them, like we know what this one looks like, we know how the fluorescein dye should look, and what can happen is a piece of this one gets translocated somewhere else. Would that be a problem? And we know that some translocations indicate cancer is going to be probably in the future, or heart disease, or other types of things, right? So, anyway, this is a karyotyping. Does anybody ever had a karyotyping done on their child, or what's that? No? Yeah. So with your child, you had a karyotyping done?
With my first one.
With your first one?
We
Okay.
determined that the child was having Down syndrome and a deformed heart.
They did diagnose that? Is that what you said? So has that done okay? Has that,
I didn't carry the term.
you didn't carry your term. So you use that information to help make decisions, sometimes really tough decisions. It can help you with those kind of decisions. And you have known somebody who had a karyotyping done?
I wasn't raising my hand.
Oh, I had one with my third child because they wanted me to with the first because I was 35 having the first. And you know, with age comes some things that sometimes you need to be ready for or help you to make decisions for. So, but I didn't with the first or the second, but the third I was thinking I better be ready because I got like a 1 and a 0-year-old at home. So I better be ready. So I did the amnio with the third. And it's a procedure that can really give a lot of information. And the genetic counselors are great. So, you know, the genetic counseling can be great with that too. And so these can be used for a lot of things. They can not just be used for embryo and fetuses, they can be used for adults. So adults sometimes come in and they are having some issues. They may be having fertility issues, or they're having diseases that they're trying to think of, and they think we need to look at your chromosomes. And so adults can have the karyotyping done for diagnosis as well. And again, when we do karyotypings here in the lab, we'll probably do that Thursday. I'll let y'all do karyotypings Thursday because we don't run out of time today to do it, but because I want to get to microscopes. But you all will see some disorders in adults that can be diagnosed. Yes?
I had a CBS when I had my son because I was pushing the 40, and it came out just fine. But I've been through a true miscarriage, and then I had this term for it. I can't remember the name of the term, but it's when your body thinks you're pregnant, but you're not.
Yeah.
And you will eventually lose it.
Yeah.
Then I had shingles, and I was definitely trying to get and have a CPS done.
Yeah. And so sometimes too, if other blood work or something comes back and they think there could be risk for whatever reasons, they'll recommend. So CBS has gotten a lot better than it was when you had your son, but when you had your son, there was high risk to CBS testing. So
I went to the first time I was going to have it done was when I was pregnant, and that's when I lost the child. But they told me at NCB, oh, there's nothing to it. You can guard yourself, no big deal. And so when I had it done down at Duke, there's a uncle I worked out for many years before. I said, oh, by the way, can I drive myself? And he said, no, ma'am. You have a spontaneous abortion. I said, I thought it was a little bit more important than what I had been told. But anyway, yeah.
well, yeah, I drove myself with him. I was by myself, and it was a sleeting storm. Okay, so that was happening from Charlottesville back then. So anyway, it's fine. But it's all the who does the procedure is what it is. It's who and they know what they're doing and how many they've done. But I did want you to know what a karyotyping is because we're going to get back to it. So guys, now I want to talk to you about inheritance, and I'm going to do a little introduction here before we go to the lab. We are going to the lab today after a little bit of a break. But here's a little introduction. What do you all know about inheritance genes and whatnot? You all know that if you say gene, I told you that's the same thing as saying I'm having a recipe for what? A protein or trait or characteristic, right? So that's what we mean when we say gene, right? And you all have heard of some things for genes. You said that you know that y'all already know that sometimes you can have dominant or recessive. What does that mean? What does dominant mean?
What you have expressed.
What's expressed? What else? What are some things y'all know? What are some things that else you know? Dominant. What does dominant mean as a trait?
That it's more likely in the population.
More likely in the population? That's a myth. That's not accurate. But I'm glad you said it. Most people think that. Most people think that. So that's an inaccurate statement. But okay, so y'all know that's not really true. So what else? Stronger, which is inaccurate. Some dominant traits are lethal and they're immediately lethal. So it's not stronger. It's not predominant in the population. Those are myths and inaccuracies. So let's let those go, okay? And we're gonna let those go. And we're gonna learn what it really means. All right. So what it really means is that what dominant means is that you have a recipe for a trait to be expressed, and it will be expressed. Now, you told me for your autosomes, how many at least chances do you get for each trait? You get at least 2, because you got one of these from your, and one of these from your, did he? Okay, all right. Yeah. All right, so exactly. So for a dominant trait, it just means that one of them gave you information for a protein to be made and a recipe to be built, okay? Recessive means that you don't have the information for that normal trait. You don't have it. If you don't have it, can you make it? No. So is everybody good with that so far? Now I want to give you the term allele, and I want to give you the definitions because I think textbook definitions can confuse the hell out of you. So I want to give you the definition for alleles. So I'm going to go back to this picture. Alleles means, if you know the alleles, it means all of the different places on the DNA that contribute to that trait. So that's what alleles mean. It means every place that would maybe contribute to that trait. So let me say this. There are some traits that are very specific. They're only on a very specific loci. So that means that their alleles are just here on the 8th pair of chromosomes, just right there, right? But some traits have a lot of alleles, which means loci that contribute to the trait. So for a trait like cystic fibrosis, do you think there are a lot of alleles, or is it pretty specific? It's pretty specific. For a trait like sickle cell, are there a lot of alleles, or is it pretty specific? But traits like hair color, eye color, skin color, height, what do you think? There's a lot contributing. There are a lot of alleles, meaning there are a lot of locations, loci, that contribute. So traits that we get this kind of rainbow effect, there are usually a lot of alleles that are loci that are contributing to the trait. You get what I'm saying? But for some things, it's pretty specific, right? All right. So here's what I want you to know. Do we really care what somebody's earlobes look like, whether they're attached or unattached? Do we really care that somebody has an attached earlobe or a lobe that hangs down that's unattached, attached? Do we care? No, but is this a trait that's inherited? And so is it this, can we use it to explain how other things that we do care about are inherited? Yeah. All right. So I want to give you all, and this is a trait that's in the autosomes. And for the autosomes, we said that means what? Your self chromosomes, right? That means that the traits that are on the first through the 22nd pair. So guys, when we talk about a trait, we're just talking about one trait. We use one letter. And it doesn't matter which letter you use. They used a D. But it doesn't matter which letter you use, but use one letter. So I am going to use A just because a capital A looks really different from a lowercase a, right? I don't know if y'all can tell, but I have detached earlobes, which are actually a dominant trait. And to recognize dominance, we use capital letters. So do I have that? One of my parents must have given me this trait because it's expressed, and I had. Come look at them if you don't believe me. Okay, really, I have this. But didn't I get potentially a recipe from another parent, the other parent, about this trait? So I might have gotten the same recipe. I might have. Would I have had to have had it to have this? Or would I have just had to have had it from one parent? Okay, so I might actually not have gotten that, right? I might have gotten no recipe for that trait from the other parent. Wouldn't I still have these earlobes?
Yes.
All right, so here's what I want to tell you about. When you use lowercase, you're talking about recessive for that trait. We're only going to look at one trait at a time, so we're going to use one letter. So you might be talking about recessive or dominant, but for these particular things we're going to talk about, you get two chances at them. So you might have inherited something from your mom that's different from your dad. This is called, by the way, remind me what the first cell was that you were called? This is called a heterozygous condition, because hetero means what? Different. Heterozygous means a different set of information that zygote had for that trait. Is that right? Where if maybe I inherited this information, same information that I inherited from my mom, I inherited it from my dad about my earlobes, I would be called what? Homozygous. Homozygous. Homozygous, because homo means what? Same. Homo means same. Hetero means what? Different. That's what those prefixes mean. So homozygous or heterozygous, right? That's all that means. So if I had two big A's, don't make me write that back, but if I had two big A's, I'm called homozygous dominant. But suppose I look like this, what would I be called? Homozygous recessive. Suppose I look like this, what am I called? Well, you just say heterozygous because it's understood that one of them's dominant, so will dominant be expressed? Yes. Guys, if you don't understand the terminology for genetics, that's your first thing to understand, or you're going to be lost. Okay? So you have to get this terminology down. Now, what I can see, I know for this trait that I got two sets of information for it, because I know that's what inheritance is talking about. We're sexually reproducing organisms. I see from the karyotype that I have pairs of chromosomes, don't I? One came from, and the other one came from. By the way, each chromosome has a homologous partner. Homologous means homo, same, chromosome, that's a partner. That's all that means. Homologous partner means that our chromosomes are inherited in partners, aren't they? Because we get one from our wear and one from our, and I do this to death because I need people remembering that. So look, Scott and I, y'all don't know Scott. But anyway, Scott and I. So when I met Scott, you think he cared about my earlobes? No. So anyway, and do you think I cared about his? No. But okay. But could he have seen and noticed my earlobes? Yes. And guess what? I did mate with him. Whenever you see an X, it means mating. Guess what? Kind of earlobes he has. He's got just like mine. He's got dominant too. Not that I cared. But okay. So I could see it though. I could see his, and if he'd been paying attention, he could have seen mine. Okay. But anyway, what you can see is called the phenotype. Pheno. You know, I don't usually spell it for you, but there you go. Phenotype. Phenotype means what you can see. Nicole, are you recessive for that trait? No, I don't know. I can't. I'm trying to see if somebody's recessive for earlobes. Are you, Justin? Who has attached earlobes? Anyway, what you can see is the phenotype. What you can see is the phenotype. I was phenotypically dominant for this trait. He is phenotypically dominant for this trait. But guess what we ended up having? A couple of children, and he's really the father. I would have no reason to be lying to y'all. So look, so anyway, he is. So but guess what we had? We have a couple of children that are, guess what? They're recessive. They're homozygous recessive. Phenotypically recessive, homozygous recessive. And that means, do I know what's here now?
Yes.
And do I know what Scott is holding?
Yes.
Because he had to give, when he got to give half the information, didn't he? And I got to give half the information. And so because we have two children that are recessive, phenotypically recessive, if somebody is phenotypically recessive, do you know both sets of information? You have to, because if they had the dominant once, would it be expressed?
Yes.
Yes. Do you get that? So because we have two children that have this, I know now what we have on both locations. This, knowing both locations, is called the genotype. So by having children with somebody who was this, do I know my genotype now for that trait?
Yes.
Yes, I know my genotype. Do you get that? You all understand that terminology? Mostly, I just want you to be understanding the terminology, okay? So a phenotype is what you can what?
See.
But a genotype means that you know what? You know all the information. You know what's from both parents. You know it. If somebody is phenotypically dominant, do you necessarily know their genotype? No, because they may either be this, but wouldn't they also be dominant if they had this genotype? So you don't know their genotype if they are dominant. But if somebody is recessive for a trait, must you know their genotype too? Okay. So here's what I want to ask you all. I want to ask you all something. Because this is, and you won't have to do this Thursday, because I know we've had a lot of information. But this is what your goal is by the time we get to visit this back again next Tuesday after the test on chapters 1 through 3. So not for this Thursday, but I want y'all to think about this and see if it kind of makes sense. And actually just try to listen and see if you can do this. Because you will have questions like this from chapter 4. I'm going to give you all some scenarios, and it means you would have to be able to answer these questions by doing certain things, but mostly understanding the language. If I told you that a homozygous dominant mother mated with a homozygous recessive man, which you all know how to write those, what would you say? Capital A, homozygous dominant, this is the mom, mates with, we said that's mating, right? With and maybe I wouldn't have said homozygous recessive, because maybe I would have just said phenotypically recessive. But if something's phenotypically, seeing that it's recessive, do you know the genotype? How would I write it? Lowercase a. And I would ask you for this trait, and this trait might be whatever, just anything. But if I were to ask you for this trait, maybe it's sickle cell, maybe it's cystic fibrosis. Maybe it's, whatever. But I were to ask you, with this mating, what's the probability that they have an offspring that has the genotype that's phenotypically recessive like the father? Could you answer that? Yes. Okay, let me tell you how you do that, and I know some of you already know. But how you would do that is by doing something called a Punnett square. So I want to just briefly show you that. Now I know most of you wouldn't have to do a Punnett square for something simple as this, but doing the Punnett square is always a good way to check yourself. So let me explain to you how a Punnett square is used and what it's used for. A Punnett square is used to predict the probability of the offsprings genotypes and phenotypes. So to predict the probability of the offspring's phenotype and genotype. Let's say this is sickle cell disease, okay? Let's say this is sickle cell disease. And does this man have sickle cell disease? Because it's a recessive disorder, so yes, he does. He has sickle cell. Does this woman have sickle cell disease? No. No. But what would be the probability, maybe, of saying would their offspring have sickle cell disease? Or what would be the probability of some other things too? We can do a Punnett square. This is always how you do a Punnett square. Ready? For one trait, there will always be 4 potential probabilities of genotypes. There will always be 4. For one trait, each one trait, there will be 4. This is the square. All right. Now, the way you do this is you take the mom and you're forgetting the dad for now. You take the mom and you think these are her chromosomes. Remember the karyotype? Remember the karyotype, the picture? This is an actual chromosome. We're just looking at one trait on that chromosome. This is the actually homologous partner of that chromosome. We're just looking at that one trait. That mom gets to give as an egg. She gets to give either this one or she gets to send this one. Is that right? As eggs. Now the information is the same for that trait, but she gets to send, when she makes eggs, it's 50% chance this one will go and a 50% chance this one will go to the egg. You good? Okay, now let's look at the guy. The guy gets to make sperm, right? And there he's got this. He can either send this one to the sperm or he can send this one to the sperm. Which all agree? Because these are actually physical chromosomes that we're representing with these letters here, right? Go back and look at the karyotype. That's the physical chromosomes. So the probability that this egg met that sperm, this is a probable genotype of a child. So inside these boxes, I want you to realize that this is a probable child, okay? If this egg met that sperm, this is a probable child. This egg meeting this sperm. Are y'all with me? This egg meeting that sperm. There are 4 probabilities that can actually happen. Now, I can answer questions. If I've done this and I know how I did this, I can answer questions. If the question says, what is the probability of these 2 people mating that their child would have sickle cell disease, what would you say?
0.
Why would you say 0? Because this child has one normal information for him, and that's all they need, right? So none of the probabilities are this. It would be 0. No child between those 2 matings would have sickle cell. Would that be good information to know as a parent going in? When you have a husband who might be in crisis a lot of the times, in and out of crisis, and you think, oh, my child did this. Wouldn't this be good information to know? Now, wait a minute. Suppose I ask the question, what is the probability of a child being a carrier for sickle cell trait? What would you say? 100%. 100%. It's 100%. That child's going to be a carrier, which means that child, whether it's this child, these are different children, by the way, but they all are heterozygous for this trait. So each one of these would have a 50-50 chance of sending this one or this one. Right? Okay. Any questions about that? If I were to ask you about a heterozygous mom, how would you write it? A heterozygous mom for a trait mates with a heterozygous male for a trait for, let's say, sickle cell. We could also use this for, it's the same inheritance for cystic fibrosis. It's the same inheritance for any of the autosomal recessive disorders. But if I told you that the parents were heterozygous, how did you write them? You wrote them this way. This is one parent mating with the other parent. Is that what you said? If I were to ask you for an autosomal recessive disorder, which means that they have to the child would have to be homozygous recessive to have the disease. If I were to ask you, what's the chance of them having a child, could you tell me?
Yes.
And what would you want to do just to be clear about it? Make sure, because this is a very serious thing you're talking about, right? So you would do a Punnett square. What's the potential for this egg? And usually just because of bookkeeping, if there is a dominant, we put it first. What would the other 50% of her eggs will look like what? 50% of his sperm will be? And 50% of his sperm will get the what one? All right, can you fill in your Punnett square now? This egg to that sperm. What is this child?
Dominant. And non-homozygous.
Wait, but it's dominant, but it's homozygous dominant, isn't it? Homozygous dominant. What is this child? What is this child? What is this child? Is two parents perfectly healthy, no disease, cystic fibrosis, sickle cell, whatever disease you're thinking of, no disease. What are you talking about? What's the chance that their child might have the disease? 1 in 4, which is what? 25%. I have seen families, perfectly healthy parents, mating 4 different times, and all 4 of the kids had it. They beat the odds. Remember, the Punnett square is for each mating. It doesn't mean that one child will be this, 2 children will be this, one child. It doesn't mean that. This is probability for each mating. And they beat the odds, and all 4 children had cystic fibrosis and early deaths. All 4. But for each mating, this is what their probabilities were. If that egg met that sperm, no disease. What would be the probability that their child would be a carrier? Because 2 out of the 4 are carriers. What's the probability that their child would be perfectly normal, no carrier, no disease? 25%. Are you good with this? What did you use to predict probability of genotypes for offsprings? What did you use? Are you going to have to, not Thursday, but by the next time we kind of, or are you going to have to figure? And all this really takes is just knowing the language. And if you know the language, you can just carefully do your little Punnett, put your parents, do your Punnett square, and answer the questions, right? But this is an introduction. So how do you feel to genetics? Introduction to, you feel okay?
Yeah.
All righty. All right. Do we really care about hitchhiker's thumbs or widow's peaks or earlobes? But are traits all inherited by chromosomes? And are those homologous chromosome partners, do we understand how they're inherited? Yes, we do. And for the autosomes, which are just through the first through the 22nd pair, for the autosomes, how many chances at least do you get? 2. Now, if it's talking about a trait that has many alleles, it means that there's going to be a what? There's going to be a rainbow kind of effect. Skin tone has several different chromosome pairs that contribute to skin tone. I have probably some of my best friends from college are medium tone, darker people, maybe like Janae tone, and they have a child that is incredibly light. But is that a chance? Is that a probability? Absolutely, because they were holding, recessives, but as they shuffled out, then that's absolutely true. That can happen. And he was really the father too. Okay, so I make jokes about fathers and really good fathers, but yeah. Moms know. It's hard to deny that when you're in there pushing, right? So anyway, any questions for me before we take a break and go to lab? No? Are you so clear? That's what I am too. All right, so here we go. Guys, how much of a break do you want? 15, 30 minutes. What? Well, I need 15. So wait a minute. So what did you say? How much?